All right. Hi, everybody online and in the room. Welcome to our distinguished seminar series at Archbold. My name's Betsy Boughton, and I'm the program director of Agroecology. And today I have the honor of introducing Dr. Chris Wilson. He's an assistant professor in the UF Agronomy Department. And he works on broad topics such as monitoring ecosystem services in agroecosystems, how pasture management affects carbon sequestration, and designing more resilient crop production systems. And he received his PhD from UF in 2017, and he was an Archbold intern in 2010. So that's when I first met Chris when I was a graduate student here. So it's really great to welcome him back to Archbold and to hear more about what he's doing. And in this very relevant topic to us, managing grazing lands in a world of change. So thank you very much, Chris, for joining us.
All right. Well, yes, thank you for that introduction and for the invitation to be here. It's an honor for me. Last time I spoke at an Archbold seminar, I was doing my intern exit seminar. I was in the old building. This is very nice new facility you all have here. And back then I was talking about clipping grasses and ecosystem responses to that. And then today I'm going to tell you about grasses and grazing and clipping. So I can't get away from this topic. Yep. I'm supposed to be in the frame for a little while. I tend to move around a lot, so I apologize in advance. Let's see. Maybe these keys. There we go. Very good. Okay. The big picture context for a lot of my work, what I'm ultimately trying to make kind of a contribution to as a scientist, what I think about as a human on the planet today is, of course, climate change. And so there's a lot of things about climate change that are very urgent and have to do with very recent changes, but I think it's always good to zoom back and look at the bigger picture. So from that perspective, I really like this figure from NASA that shows, based on ice core reconstructions, other proxies, CO2 in the atmosphere the last 500,000 years. You can see glacial, interglacial transitions associated with shifting CO2 between about 180, 270 parts per million over the last ice age. And then, of course, in the industrial era, we begin a large planetary engineering experiment that is sort of like a moonshot. That's what I think of when I see that trace of our atmospheric CO2 put into this bigger picture context, right? So, of course, this is an experiment we probably ought not to be doing, but I do think kind of helps to look at this big picture. Let's see. You're gonna have to hit enter. Okay. I don't know how many of y'all have been following this. This is now like much more recent climate stuff. We're starting to see, I think, some of the major impacts that are going to unroll if we continue on this moonshot that we've been trying for. What you see here is the sea surface temperature anomaly. So it's the global average temperature deviation from a 30-year baseline like trend projection. And what it's plotting is basically we've got from 1980 to 2024, you see a long period of time where, so these are the earlier in the 80s, this is getting us up to contemporary observations, oscillates, there's a general increasing warming trend. 2023, something dramatic happens. And we still are seeing a lot of debate and controversy in the climate science community trying to explain exactly why that's happening. El Nino is part of it, but can't account for all of it. Some of it is probably reduction of aerosols. So we've cleaned up our act with ocean shipping. That's probably led to additional heat actually gain in the ocean. And then 2024, we are starting higher than we did in 2023, yet to be seen where this is going to settle out. If you combine this with this surface temperature globally averaged, we would already be like close to 2C of warming if this persists. So it better not. Anyway, I think it provides a little bit of urgency to what we're going to talk about. So I'm going to discuss some ideas around ecosystem management for climate mitigation, but I want to put it in the context that none of this should be construed as we're going to solve climate change with ecosystem management. We absolutely have to do that decarbonization. We have to get off that fossil fuel driven trend line. The ecosystem management part comes in and helps us ameliorate a little bit, helps us draw down CO2 a bit, buys us some time, but it should never be thought of as a substitute for decarbonization. So I'm just putting that as a general disclaimer for the background here. I'm going to try to talk about three different things, time permitting. First thing, I'm going to do a deep dive into a recent paper that I helped co-author that really digs into how to understand some of the nuances of how grazing plays out, and thus hopefully provide a more interesting framework for thinking about the impact of grazing management on carbon sequestration, hence the potential for climate mitigation in global grazing lands. Then I'm going to pivot, talk about some generic challenges that we have in actually quantifying soil carbon at scale and thus forecasting changes in soil carbon. So if we want to foster this ecosystem service, we need to be able to document and monitor it. We'll talk about some of the issues why that's actually really hard. Then I'm going to make a quick plug for process-based models, time permitting, that will have some actually new data in it. Okay, so for part one here, why talk about grasslands? Many of you, if you've heard people from the ranch talking, may have encountered this before, but globally speaking, pasture land and rangeland together account for about a quarter of the ice-free terrestrial land area. It's a very large land use that is basically already under human management in the form of livestock grazing. So if there are ways to better manage that activity that do in fact have implications for soil carbon, it's potentially scalable over a large land area. Yeah, and so it actually turns out to account for a similar amount of the global total of soil organic carbon, so around 25%. And to put that in context, if you decomposed all of that into CO2, that's worth about 50% of the atmospheric pool of carbon. So relatively modest changes in that pool could have decent implications for the atmospheric CO2. There's a lot of different ways we can think about for trying to increase carbon, foster carbon sequestration in grazing lands. A lot of things getting a lot of attention, right? There's direct additions of organic material. We could try to put out compost, biochar. There's some interesting work on that. There are challenges to scaling that over large areas. Nevertheless, I think that's an interesting and important research direction that we may have underdone a little bit in Florida. Silvopasture is another interesting land use concept, basically stacking trees on top of pasture. Of course, there has to be an argument economically somehow to do this, but the idea there is you're building a pool of woody carbon biomass on top of your soil carbon. Enhanced weathering, right? This idea of you get basaltic rocks, they react with CO2 and form carbonates. This is getting a lot of attention in like society journals, a lot of interest in this method. It's potentially an interesting alternative to liming because you actually lock up CO2 rather than release CO2 as you're ameliorating your soil pH, as you would with a liming practice. Management of these ecosystem processes of fire and water, big deal in Florida, big deal in a lot of grazing landscapes, lots of interesting work that remains to be done. I'm going to take a deep dive today into this grazing management piece, again, because the livestock are there, they're playing a crucial keystone role in the ecosystem, and they're kind of the main human interest in these ecosystems. So their management is potentially a very scalable thing to address. And indeed, if you start looking at what third parties who are getting into the voluntary carbon markets say that they will pay for, improved grazing management is on that list for ranchers. So just as one example, Agoro has this. I spoke to them and many others conferences. I think it's very interesting that they put that in there, along with increasing species richness and fertilization practices. We're trying to get some good data in my lab on the bottom two. The top one I think is really actually kind of challenging, but I'm going to talk more about that. I think we need to build out the scientific basis for all three of these much better. That's just my opinion. So a lot of this is going to kind of summarize and synthesize a very recent paper that I co-authored with my friend Paige Stanley and the group that she's with Francesca Catrufo at Colorado State University, where we tried to unpack why is it that the literature provides so many conflicting insights about how grazing actually impacts soil carbon. Sometimes it's like it's a negative factor, it's positive. all over the place. And so basically just trying to synthesize what do we think we know about this. So what we did was we bin things into kind of the three major planks. You've got animals, plants, and soils. Let's unpack our best understanding of each of these and then see how we can fit these together in terms of a framework. So we're addressing grazing pattern. And so trying to add nuance to this. A lot of work on grazing consists of dichotomous contrasts, ungrazed versus grazed areas, including my own. I'm just as guilty of this as anybody else. And then when we do get into some of the subtleties of how grazing is playing out, we're typically just adjusting the overall stocking rate or that overall like grazing pressure, quote unquote. Different ways to quantify that, by the way. But there's a lot of other aspects that could be important. So we get into that as well. Then the key thing here is that all of the carbon sequestration potential comes from the plants. They are doing photosynthesis. That is the net sink of carbon, right? And so at the very least, the ceiling of what is possible can be inferred almost directly from the plant ecophysiology status. And so we wanted to look at what are some of the key factors of plant stand ecophysiology by which we think the effects of grazing are mediated to then impact soil organic carbon. Then within soil biogeochemistry, there's a little bit of a story there. I'll tell a bit of it that kind of shows like in the last 20, 30 years, we've really been updating our understanding of how soils actually cycle carbon. So we tried to put that together with these other pieces. Okay, so just getting to the sense, we use the term very specifically grazing pattern to suggest that it's not just stocking rate that matters. We hypothesize that other aspects of grazing pattern are likely to have important effects, either because they're impacting the plant ecophysiological status or perhaps the soil directly in some cases. So you can think about grazing is actually this really complex and nuanced interaction between animals and plants. If you've gone out to the ranch and you've watched cows actually interact with plants, they're not just like lawnmowers that go out and uniformly cut everything, right? They have behavior, they have culture that's transmitted. They have things they like, things they don't like. They're selecting from parts of the canopy, right? They're grazing, it's like top down rather than just like this buzzing from underneath. So there's a lot of complexity to this. So basically, quantifiably, we think at the least you want to take account of frequency, timing, duration, and intensity. The frequency is how often a given area of ground is exposed to that grazing activity. That could be infrequent or frequent. All these are on a continuous sliding scale, yeah. Timing, that's a really interesting one. You could have the exact same number of animal unit days of utilization distributed in the spring versus in the fall. Depending on the phenology of the plants that you have, the impact on plant composition is likely to be quite different between those choices. So timing likely to be important in shaping those plant communities. Duration, that's how long the animals are remaining resident in a given area during a grazing event, right? And this is going to be related then to frequency if you hold stocking rate constant, right? Because if you're moving more frequently, by definition, you've got a shorter duration. Yeah. And then intensity is more of an instantaneous thing in this framework where it's how much of the available biomass have you utilized during that grazing event. So that's kind of the way that we propose to quantify that. So that's kind of the sum. All of grazing would theoretically, we want to study this. It's really, really hard. And of course, we can't do factorial studies where we attempt to isolate all of these and study them, but we do have to get a little bit more nuanced in some cases, I think, to really understand why is grazing having one effect in one place and a different effect somewhere else. Key aspects of ecophysiology that we propose. Some of this makes a little bit more sense in a rangeland context, but they all kind of apply everywhere to some degree or another. Overall canopy cover, so how much bare ground you have, right? That's obviously going to be very important, both directly in terms of how much photosynthesis you can do, but also how stable that soil is, how susceptible to erosion, things like that. The overall productivity or primary productivity, right? That sets that ceiling. You can only sequester what you have initially fixed. But that initial fixation is often only very loosely correlated to the amount that you sequester, because then it turns out that how it gets allocated and into what types of tissue pools is really important. So allocating to roots has much different impacts than allocation into shoots. We know from a lot of work over the years that carbon that gets deposited into the soil from a root system tends to more efficiently promote the buildup of organic matter than a comparable amount of carbon that gets deposited from litter deposition from shoots. So how much root below ground allocation you sustain likely to be very important. Unfortunately, it's very difficult to quantify. It's part of why we have a lot less data on that. Um, Quality of the inputs, that matters. I'll come back to that. Then, of course, the overall diversity. So this is going to have direct effects, but also effects like the diversity of litter and root systems impacting a soil shape the microbial community composition. And in some cases, this is probably very variable, but in some cases, that can then impact their physiology and the efficiency of the microbes, which then can affect the soil carbon stocks. I'm going to get into the biogeochemistry a little bit here, and just quickly tell this story. We used to think of soil organic matter as remaining resident in the soil, largely after having formed these things called humic substances. So you see a lot of old school literature talking about the buildup of humus in a soil. This is then related to like how much complex organic inputs you're putting into it. So this is this humification idea here on the left. On the right is a related concept called selective preservation, which suggested that the more complex molecules that you put in, they're harder to decompose. It's harder for enzymes to find sites to bind and then start to initiate that breakdown process. So therefore, putting in a bunch of like tough cellulosic material is the way to go, right? We basically think that this either is overstated or in some cases maybe entirely backwards. And there's a much bigger importance that has more recently been placed on the role of the microbes themselves. So it turns out that the microbes in the soil are not only decomposing organic matter, which they have to do to sustain themselves, get their own energy, that mineralizes nutrients, a lot of important benefits, but they actually themselves contribute to the buildup of soil carbon. So it's both the microbial necromass, the dead microbes themselves, as well as byproducts of their metabolism, that is capable of entering into stabilized associations with soil minerals and contribute to some very long-term pools of organic matter in the soil. So in this paper from Josh Schimmel, his group, they refer to this as an entombing effect. I think is very, I like that language. I don't know why. It's like the Egyptian mummies of soil microbes or something. This puts a central role then on microbial anabolism. In other words, the buildup of microbial biomass is thus likely to be important in your ability to create this stabilized soil carbon. So then the conditions that foster more efficient microbial anabolism are likely to be very important in explaining variations in carbon stocks. So in the paper, we kind of take a variation of this idea that Francesca developed and published 10 or 12 years ago, this so-called MEMS hypothesis, the microbial efficiency matrix stabilization hypothesis, which puts a big emphasis on a distinction then between particulate organic matter and mineral associated organic matter. So in this framework, mineral associated organic matter are those molecules that have entered into associations with those mineral particles. This has to do with net charge relations typically, so adsorption effectively. And then particulate stuff, this is basically like very advanced litter decomposition products. So like if you imagine a fragment of a root or something that's been decomposed for many years, you're going to see the remnant of that as POM or particulate organic matter if you sieve out soil. So this is kind of a loose framework. Works pretty well in loamy soils. I think it applies, but with some more nuance in Florida systems. We can't do simple size fractionations to get at this the way that seems to work reasonably well in loamier systems. But nevertheless, I think it's a useful thing to consider here. So the key then is we want to put these pieces together. And so what we've kind of linked then is, okay, you've got a grazing pattern on the top, filtering through the outcomes that that has for the plant community in the middle, to then hypothesized effects on these allocation, ecophysiology elements, and then what that's going to do to the total soil carbon stock, and then that balance between the mineral associated and the particulate forms. So this is kind of all like a conceptual synthesis, but also a springboard for hypothesis generation. Okay. Um, I'll take it kind of incrementally here. So at the top, we start with a scenario that we will refer to as undergrazing. And so just with the implicit understanding here that this is a pattern that is integrating over those components of intensity, duration, frequency, and timing, right? So it's not just stocking rate, it's how that grazing process is played out, where you basically very minimally touch the plant community at all. So you're going to have high canopy cover, but if you let plants go really tall and accumulate lots of standing dead and lots of litter, they start shading themselves, which starts to put a clamp on productive potential. This starts to favor competition for light, which will shift phenotypic expression to favor shoot as opposed to root growth. You're going to have tissue that is on average older and more senesced. So you're going to have lower quality inputs that are less efficient at promoting microbial growth. And in some cases, you may actually lose diversity, right? So in some of these grasslands, and we even look at some of the wetlands, right? We remove grazing, you think it's going to be a great thing, but then actually plant richness declines and you don't have the composition you wanted anymore, right? So a lot of these systems, carefully regulated grazing seems to be important for maintaining that diversity. So we speculate then, okay, you're probably going to have some kind of moderate outcome on soil carbon favoring the particulate pool. What we would consider optimal grazing, what you're trying to get at here is just any integrated grazing pattern results in high canopy cover, high productivity, right? Avoiding that excessive accumulation of self-shading components, maintaining a robust below ground allocation pattern, maintaining target diversity and composition. We think that this is a scenario which should favor soil organic carbon the most, and probably specifically from the mineral associated forms. Overgrazing, we've all seen this. Highly overstocked sites, poorly regulated grazing. The grass never has a chance to grow beyond like a tiny little nubbin. You have seriously kneecapped your ability to fix carbon in that kind of a site. So you're going to have low productivity, low canopy cover. The grasses are spending all their time desperately trying to reestablish leaf area, so they're probably not doing a whole lot of below ground allocation in that sense. Input quality could be high, could be low. It depends really heavily on the context of what species you're working with. You're probably at that point down to the last survivors who can handle this kind of abuse. So your diversity is lower, and we think this is going to result in a low soil carbon. So just kind of put these pieces together. There are different ways you can kind of trace different putative pathways through those layers that I think could be interesting for hypothesis generation. I would say, for really simplistically, if you want to really optimize for carbon, you are probably talking about trying to maintain on average as dense, leafy, vigorous, reasonably diverse stands of plants as you can out there in your grasslands. I think this is good news personally because it makes it a little easier to kind of target what it is you're probably looking for. Specifically, we would suspect, if this framework is at all correct, that what you want to avoid is patchy over and undergrazing. In other words, the scenario in which you've got grazers out there and they're way overutilizing some patch, completely ignoring other patches, and your whole landscape is basically on either side of that. You spend very little time in the middle, in that more optimal zone. So you can't get rid of that entirely, right? This is all going to be on a spectrum, on a kind of patchy mosaic, right? But you're trying to get as much as you can more towards the middle. I'm personally very management system agnostic. I know my colleagues work on a lot of things like adaptive multi-paddock systems. I think those are great. You do them. You can get to some really favorable outcomes with a variety of approaches to management. So-called set stock systems could work pretty well, especially if you can vary your stocking rate over the year. So if you have the latitude to add animals in when your growth rate is really high and then take them out when it's lower, those kind of systems could actually work pretty decently well. So, and I'm also very fascinated by pyrrhic herbivory as well. I think there are lots of ways you can kind of move towards this. So that's kind of that feel for that part of this. What I want to pivot to now is, suppose you are interested in implementing some practices, whether it's grazing management or some of these other things on the list, to try to accrue more carbon at your site. Maybe you want to participate in a carbon market type of scenario. There are a lot of people doing that. Again, I mentioned this before. I have been assured that this particular company is now open for business. Hypothetically, people in Florida could apply for this. So I was told. I don't want to speak for them though, but that somebody should follow up on that. I'm going to bin this out. I think there are two major challenges why actually quantifying soil carbon and then its changes over time at the requisite scales is really hard. And then I think there are some directions we can take to move towards some possible solutions to that. So the first big bin here is this idea of model structural uncertainty, kind of reflecting again on that shifting paradigm that we have for the underlying biogeochemistry, the underlying understanding of how carbon is cycling through the soils. I'll elaborate on that a little bit. And then we have this problem of how very variable these systems are. And I think everybody who's gone out and collect soil cores, you can kind of get this intuitive feel for this, that you can see patches of ground that look really similar, and there's like, man, the soil looks way different here than it does there. I have no idea why. At least we feel that way a lot. So this contributes to what I would refer to as measurement or sampling uncertainty. All right. So the soil complexity part, I told you a little bit of that story. Another piece of context that I think is important to bear in mind here is that the soil is actually itself a complex below ground ecosystem. It's not just a repository of carbon, right? A place where you can shove carbon to go in and lock the door and then come back later, right? All that carbon that's going below ground in the inputs, some of it gets stabilized for really long periods of time. A lot of it gets decomposed to support this below ground food web, where you've got microbial organisms, bacteria, fungi at the base. Then there's all these trophic layers, ideally on top of that, constituting a quote healthy soil. Yeah, the soil is also a very physically structured and intricate type of environment, particularly in systems where you have a little bit more clay than we tend to have here. But even sometimes in surprisingly sandy sites, you will see this type of what we call aggregate structure developing. And so that's at different scales, soil being glued together by the organic products of the microbes themselves or being secreted by the roots. And that forms this spatially structured environment that has everything to do with the maintenance and activity of that soil food web, and also impacts on the cycling of carbon through the system. So just like with the climate change, zooming out to the big picture, to me, this is kind of the big picture that's always worth bearing in mind with soil. We're not just doing one thing with it, it's doing a lot of stuff with it. So we've got this shifting understanding of the soil biogeochemistry, a renewed appreciation for the big importance of microorganisms, a lot of work going into characterizing these different stabilization mechanisms that have really big implications for the residence time of carbon in the soil. And then we go to the task of forecasting and monitoring at scale, for which we almost always are going to require models. And the most widely used model in the world doesn't have any of that in it. So on the left here is the century model. This is one of the long-standing kind of gold standard biogeochemical models. It is embedded in Earth system models. It's embedded in crop models. It's embedded in the Comet Farm tool that is being used by the USDA to help quantify and validate carbon credits. It works really well for a lot of different tasks. It can reconstruct observational gradients, particularly at large scales, of observed soil carbon. But you'll notice something about it. What it is it's a collection of pools. They all have a linear decomposition dynamic given by a fairly simple first order differential equation here. And each pool kind of does its own thing. It can transfer to another pool, but you'll see microbes are represented here. They're just one or two pools among many other pools. They're not playing any kind of central catalytic role in the whole process. And they're considered to be a fast pool, so they're actually not making any meaningful contribution to long-term carbon over time at all, according to this model, right? We know this is wrong, but we're in this situation a lot. Models can be really, really wrong on the science. It's still useful for prediction. Where that tends to fall down is if you are trying to forecast the outcome of interventions. I'll elaborate that in just a second. On the right-hand side here, your right-hand side, this is an example of what we call a microbially explicit model. Again, there are many variations of this, just as Sentry is far from the only first-order linear model. So just archetypal representations of these types of models. This was developed by Will Weider, very prolific biogeochemist in the kind of 2010, 2012 range. You'll see he's got two kinds of microbes. He's actually making a distinction between R and K selected microbes. So there's a little bit of community composition going on inside the model. And both of these tools are in the center of everything. So all of the inputs, these litter terms, have to go through the microbes. And then they are either making a contribution to chemically protected pool. So this would be an analog to that particulate pool where the complexity and recalcitrance of that material matters, or the microbes are making a much bigger contribution to this physically protected pool, an analog to the mineral associated pool that we saw in that BEMS framework. So this is an example of a model that's trying to more faithfully embody our understanding of the underlying science. But that doesn't mean that it works better all the time, right? Even though we think it's closer to the truth. When this is going to matter the most, I think, is when you are attempting to forecast the response to an intervention or a perturbation. Because it's one thing to kind of do curve fitting, basically, and reconstruct observational data. It's another to say, if we intervene and X happens, or we increase nitrogen by 20%, or CO2 rises by 100 parts per million, this will be the consequence in this system. For that, getting the processes, getting the mechanisms right is much more important. So as an example, Will published this in 2013. This was actually with a much simpler microbial explicit model compared to first order linear types of models where they were looking at a high-end warming scenario from the IPCC. What would be the net effect on global soil carbon stock of the warming that occurs under these different models? So there's this green line on the top, and that is the Sentry type model, merrily humming along saying we're actually going to increase soil carbon. There's a CO2 fertilization effect. Good news, everybody. The bottom dashed line is one implementation of the microbial model that says, actually, we're going to lose 250 billion metric tons of carbon because of the microbial physiology being perturbed. Then you've got a line kind of that splits the difference that says it's still microbial physiology regulating everything, but that physiology is allowed to acclimate over time. And then it kind of mean reverts quite a bit, still forecasting a loss. So I think it's my intuition that this is going to apply equally well to when we try to manipulate grazing, when we try to manipulate fertilization practices, plant composition, adding materials, anything that pushes us meaningfully outside the historical baseline that you're training your models on. I suspect that the more accurate type models, if we can represent the mechanisms that are actually playing out reasonably well, will give us better forecasting, will tell us a better story about our potential for carbon credits. Okay, so that's that first piece. The second piece that confounds us routinely is the diversity and heterogeneity in these systems. So I alluded to this before. This is a picture of some work actually that we did at the ranch. look familiar to many of you. This was a survey of the upland pasture and the kind of the interior planted pasture area, um, where we very specifically excluded wetlands, hammocks, any other land type that wasn't clearly Bahia grass, uh, grazed pasture, right? And at this point was just trying to look like correlationally at all the different landscape drivers. We were interested in remote sensing, which is why we overlaid this map of EVI underneath all this. But the important point I want you to take away from this is that there was about a threefold variation in estimated soil carbon stocks to 15 centimeters on land that looked about the same everywhere. So what accounts for that? Who knows? We can explain a little bit of that variation with remote sensing and some of the edaphic factors, but far from satisfying in my opinion. Just an example there, right? Now imagine you've got much more strong contrasts in the land cover type, the composition, how the grazing is playing out, et cetera, et cetera. So we've got lots of variation at a site scale, which is probably the scale at which you're going to attempt to monitor these carbon stocks, right? So another neat thing that Paige did fairly recently, this was the lead author on that GCB paper that was the first part of the talk, is really do the work for something that some of us have been chatting about for a while. I didn't know that she'd done this until I was preparing a talk for a conference that she happened to be at. But I found the paper like 2 days before I went. So I was like, okay, I need to cite this and make reference to this now. The idea here is, what is the impact of all that variability on your statistical power? Okay, and so they worked it out for a kind of a classical frequentist type 1, type 2 error paradigm. You're trying to assess your ability to correctly reject null hypotheses. And basically, she compiled data from 5 different range, or maybe 6 different range sites that displayed different levels of variability to have some realism in the analyses. And then they simulated the effect of a sampling approach that had 10 samples, 30, 90, or 200 samples with different hypothesized effect sizes. So what this is saying is, suppose you measured at time 0 somewhere, somebody did a practice and it's increasing the soil carbon by the amount shown on the x-axis. The y-axis is giving you your statistical power. It's unfortunately clipped off in this image, but it goes from 0 to 1. You want to be high, right, to have a good, robust design. How many samples do you need? And what you'll see is, for these small effect sizes, like at the most heterogeneous sites, so like this green line there is a very variable site. If you've only got 10 samples, forget about it. You're basically 0. You're not quantifying anything. It's literally a random number generator, what you're getting out of that. That's true for a lot of them until you get up to some of these really significant sample sizes. If you want to get power 0.8, which given the realistic effect sizes that we think you're likely to want to be trying to track, for a lot of these systems, you're going to need the equivalent of 100 or more samples. That's if you assume you have no other source of information, right? If you're just relying on sampling. But I think it's a good way to calibrate our intuition about how tricky this is likely to be. The other problem that I would point out is that if you try to make yourself feel better by saying, okay, our sampling's inadequate, so we're only going to take seriously situations where we meet a statistical significance threshold, then we're fine, right? No, you're not. If you have low power and you filter on statistical significance, you actually bake in large, what are called type M and type S errors, magnitude and sign errors. So on expectation, you will be a very exaggerated, biased estimate. So you can't afford to think you can get away with low power if you are actually trying to get, accurate quantification of like an effect that could be 5, 10, 15, maybe optimistically 20%. You really got to dial that in. Okay, so does this mean we are just doomed, that we're never going to be able to do this? The problem that you will hear a lot is that, if we insist on that, the cost overhead of administering these systems is going to exceed the value of the carbon payments. This is those kind of studies where we get lots and lots of sites sampled. That's the kind of thing that graduate students do with grant funding. It's not a particularly profitable business proposition. I don't think that we're always going to need that, but we do need to start taking this seriously, building up pooled data sets, doing the careful meta-analytic work so that we understand how much variation to expect at different spatial scales, how much variation to expect across different sites, to, in other words, set strong expectations that are quantitatively informed, that basically act as prior information in more informed analyses. The other thing we can do is make use of these process-based models as we develop them and as we build confidence in them, provided we are accurately assessing that confidence that we should have in them. Okay, so I think that there are ways forward here. There's also help that can be done, I think, from a remote sensing angle, particularly if we recall the central role of that plant canopy, that plant composition that we think sits in the middle, regulating the effect of these interventions on soil carbon. So if we can get better, more resolved insight into that linkage, then I think remote sensing will start to become more and more useful tracking the soil. I'm a little bit more pessimistic about people trying to do remote sensing of soils directly. Obviously, in tilled systems where you can get a good view of a bare soil, that might make some sense. Never going to be the case in a grassland. Ideally, you should have lots of canopy cover. So anyway, um, let's see. We've got till 4:30 technically. Okay. I'll just spend a couple few minutes on this last part and then open for questions. I want to make a quick pitch again on pushing on this theme of building out process-based models. And this is going to be a little bit of a case study looking at one way that we might start to think about linking climate change impacts to grazing lands. So far I've been talking about grazing lands as potential for mitigation. Right now I'm going to look at what does all that potential warming mean for production. So that's going to also impact mitigation insofar as, again, we've got this plant sitting in the middle of the whole thing. So I'm not going to go over too much. Many of you are very familiar with the distribution of Florida grazing lands, the fact that we're these subtropical systems. The key point I think I want to emphasize about all this is that I believe that the fact that we're dealing with these really tough C4 subtropical grasses, particularly Bahia grass, has given us a strong complacency about their resilience to climatic stressors. It's true Bahia grass is extraordinarily resilient. That doesn't mean there won't be meaningful impacts on quantity and potentially quality of production as we move into climate change scenarios. So this figure right here, these are this is the vapor pressure deficit, or basically the total evaporative potential demand. And this is pulled from the TerraClimate data system. I highly recommend playing around with this if you haven't already. This was taken for our research site outside Gainesville. What it allows you to do is look at, in blue here, is the past 30 years of observed weather data interpolated for our site. The yellow dots are showing, and this is all spread out, okay, of months of the year here, right? The yellow dots are showing what it would have been given a 2C of warming scenario, and the red dots show what it would have been given a 4C warming scenario. So getting towards that moderate end of the IPCC projections, which hopefully we don't get there. We might. What you will see is, of course, there's a non-linear increasing damage as we go higher up the warming cascade. The gap from 0 to 2C not nearly as profound as the gap from 2 to 4C. In the 4C scenario, the evaporative demand that we're going to experience will be staggering, according to this data. And it's going to come at the wrong time. We're going to see a lot of evaporative demand starting to build in March, April, and May. That's our natural dry season, even going into June. If summer rains are late, you're going to be exposed to that as well. These sites are going to be bone dry most of the time, that time of year. That's a huge impact to your forage production if you don't have an irrigation system ready to make up the gap, which, let's face it, most ranchers don't, right? It's a big investment, and then we'd have to worry about water allocation and allocating that out for this land use, right? That evaporative demand is going to continue through the summer months. So anyway, I wanted to take a quick look at, how this is likely to impact things, again, with this idea that process-based models should ideally allow us to link up and help us do parts of this upscaling. In the interest of time, I'm not going to belabor that. The way that we've started to look at this is through the lens of a fairly simple process-based model that takes account of leaf area, intercepting sunlight, the conversion of sunlight into biomass, which is reflected in this parameter RUE, or radiation use efficiency, that I have in the middle of this diagram, and then the impact that that is going to have on growth as you go over time. So the challenge from this point of view is to understand how these different environmental factors impact radiation use efficiency. And then if we've got different management options like fertilizer, like irrigation, subsidizing soil water supply, climate change driving increasing temperatures, increasing evaporative demand, potentially depending on the precipitation impacts, increased or decreased inputs into the soil water, start to work through what our RUE model suggests to result. And we can use the TerraClimate data to do this. But to get there, we need to build up our model and have it be informed by real world data. So among other reasons, we have set up a rainfall exclusion experiment to try to manipulate soil water and incidentally then link soil water supply to forage production, as well as temperature forage production. Because I was interested for a variety of reasons in fertilizer deposition, we actually have a kind of a fertilizer rate trial nestled within this. So we have everything from no fertility inputs to twice the IFAS recommended rate of input for this type of system. So we can see huge responses to fertilizer in terms of our radiation use efficiency. That basically means fertilize more, get more production. Makes perfect sense. There's a bit of a nonlinear effect, right? So there's a nice kind of middle amount of soil water that optimizes production. You can either be too dry or too wet on either side of that. Temperature is basically linear, except there's a bit of a nonlinear signal at higher fertility levels. So great, we can take this data, put it into that network. And so we propagated it through in a Bayesian network to then look at scenarios. So this is posing those intervention questions. What happens when we ratchet up temperature 2C and have different levels of fertilizer management? So starting to look at management by climate interactions in a causal process-based model framework. So based on our current field data, so this is all preliminary, we need more years, we need more sites, yada, yada, yada. Just to illustrate the principle of the thing, we can start to look at how a pretty modest warming scenario of 2C is then likely to impact RUE and tissue nitrogen. So tissue nitrogen is your crude protein, that's your quality, right? Under either 2x fertility or no exogenous fertility, both scenarios see a meaningful decline as a result of that warming. So you're seeing lower quality regardless. Then the RUE signature sort of flips. So we see a little bit of benefit of that warming to growth of Bahia. Again, this is all else equal. So I'm not talking about if we have a really bad mega drought, it's not going to be like this. But if conditions are similar, except a modest warming effect, a little bit higher under no fertility. But if we're fertilizing heavily, a shift with a decent amount of uncertainty around it, but a shift towards a lower forecast for RUE. So this is just starting to show how I see these pieces starting to fit together, ideally more sites, more data, and then start doing similar things on the soil biogeochemistry side to then allow us to kind of put these pieces together. This is the stage where all I'm doing is hand waving. Imagine them all put together really nicely. It's an aspirational goal for 5 years, something like that. Anyway, so I'll leave it with a few takeaways. I think overall, again, on that grassland SOC side, I think the platonic ideal is sort of dense, leafy, vigorous, reasonably diverse stands of plants, avoiding that patchy over undergrazing. Currently, we are significantly hindered in our ability to forecast SOC by both scientific uncertainty and measurement problems, and that we can use targeted experiments, Bayesian networks, to make some progress with using process-based models to redress some of these limitations that we currently have. So thank you for your time. We'll leave it there.
Hello. Hey, thank you. Thank you for your presentation. I have a question first. Start with the carbon pump
Mm-hmm.
in the previous. So my
Yep.
first question is, do you think the the carbon pump rate would be greater in those kind of a humid subtropical grassland, or it's more the carbon pump rate will be stronger in the like a I don't know Great Plain kind of semi-arid pasture?
So a good question. So in general, warm, humid conditions foster microbial activity. That's likely to be both anabolic and catabolic activity. So you're likely to see a higher respiratory output, a higher rate of breakdown of organic material that's accessible to the microbes, as well potentially as higher growth rates. But you would have to look at that carefully, and you need to separate out like gross from net kind of impacts with that. So in general, we see it's actually more of these like temperate type of systems that tend to have the most like seemingly long-term potential for building up that mineral associated carbon. And you see really high carbon stocks in like really high latitude systems like permafrost, stuff like that. Of course, those are very slow. That's taking place over thousands of years, right? But that's because largely the decomposition rate is so retarded by the cold frozen conditions, right? So I don't know if that answered your question satisfactorily, but
Yeah. Ask you for
oh, I see.
Scenario. So what do you think
Yep.
the let's say +4 associated
Yep.
degree effect, how would they affect the carbon pump rate?
Yeah. So the thing is, we will need to have a sort of nuanced network sort of like this. And by the way, all these interactions like can embody, it could be linear interactions, it could be nonlinear functions that link these things together, right? A similar thing for the microbial response. One of my PhD students has used that system to study large gradients in soil water impact on respiration. So she did this in a lab incubation setting. And so we're actually writing that up now. So that's going to start getting us at, okay, we know soil water has got this big effect on plants. What's it doing to the microbes? I personally think that soil water supply is kind of like a master variable for a lot of things in these ecosystems. So it'd be interesting to see, how does that then impact, potential microbial contributions to it. The problem is in our soils, we actually aren't super confident about how much of our soil carbon is of microbial origin versus plant origin. That work is still ongoing for Florida soils. So, right. Yes. Oh, I guess you got to wait for the mic. Sorry.
Thank you. So I'm thinking about that grazing box right on the one you were
Oh,
on.
you want to look at this? Okay.
The grazing box is not connected to anything.
Yep.
And
it is.
There's emerging work suggesting that rewilding ecosystems with big grazers can influence carbon processes. So could you speculate a little bit on how grazing either of livestock or wild herbivores might influence the processes you talked about, in particular ecosystems that are historically adapted to having big grazers, like most grasslands, and ones that are maybe not? We sort of sit on the cusp of where bison maybe didn't used to be.
So the impact of the grazers on soil carbon?
Impact of the grazers on soil carbon.
Yes.
The historical contingencies of the evolution of the plant communities to grazers.
So I think that's going to hinge fairly crucially on how well this kind of heuristic diagram reflects the reality that plays out at your site. When you introduce those wild ungulates, do you see a net shift towards a grassland that looks more like this than like these endpoints? If they have that net effect, I would suspect, yes, there will be an accrual of SOC in that. If this thing falls apart in that context, then who knows?
So just a question, do you shift towards that kind of moderately diverse leafy thing?
Yeah, I think so. With high levels of below ground allocation, high levels of deposition from the roots, both, I should mention it, that's both the roots themselves dying and turning over. So that's like a root litter deposition, as well as rhizo deposition as a process that's exudation of compounds like glucose and amino acids, as well as sloughing of, the cells that line the roots. That stuff actually, the exudation really supports a very vigorous microbial community right in that rhizosphere area that the root is impacting. So all of that, the sum of all of that is what matters, I think.
I'm glad we came to this framework because I wanted to ask what the role of invasives is in this framework, and doesn't more invasives mean more carbon storage, or unpalatable invasives mean more carbon storage?
Well, interesting you bring that up. Pretty related to that, there's a lot of work on like woody encroachment in the West, and that's often associated with losses of soil carbon stocks as you shift away from graminoid to like shrubby, woody types of communities. I think that's honestly going to be a case-by-case basis with the specific invasive plants that you are talking about. Whether they're going to, compared to what's there without them, move in that direction or not. Somebody should look under Kogan grass. I'd be fascinated to know, but this is, again, we're like in the big picture of ecosystem management, we don't want to become too monolithic about one outcome. So I wouldn't argue that even hypothetically something like Kogan grass, let's say we do get 20% higher SOC, does that outweigh the impacts on community composition and diversity and fire cycles? Probably not, in my opinion. So.
Well, could we go back to that nice RU diagram that you had? The
RUE?
Yeah.
Okay. This is my very sketchy in development stuff.
Well, so you mentioned,
shouldn't scoop myself too much, but there I did.
you mentioned Pyrex diversity, Pyrex herbivory.
Sure.
And
sure.
I was really thinking in terms of, I really like the idea of the entombed soil carbon, but the truly entombed is black carbon or recalcitrant carbon, where, sort of charcoal or real black carbon. And that's a big part of, say, scrub systems. And, I really wondered whether we actually have a firebox that has an even more entombed carbon.
I think that char absolutely needs to be investigated. I would love to see more like 13C labeled char experiments or something like that to really properly trace its fate in our soils. Observationally, my personal experience with fooling around with char at a garden scale is that I added a whole bunch in an area, and then 3 or 4 years later, it appeared to be entirely gone. I don't know what happened to it. I think it probably flushed down the profile beyond where I was digging. So we need to understand how, is it, how mobile is it in the soil? Does it need some clay there to hook up with and interact with to really form that kind of durable contribution? I don't know.
Yeah, thank you. And then it's really a Betsy question, and you, I think. I was curious about your rain out project, and just wondering to what extent would that cross-refer to the project Jean Charles Chu's doing, and are we producing data that would be another experimental plot for them just from what we're actually doing on the ranch anyway?
I think so. Yeah, that was a fun part of touring around today was taking a look at that. And y'all have where I've got the nutrient manipulation, y'all have that above and beyond water doubling kind of treatment. So that's kind of interesting. Because I think your data would be interesting to cross compare with my 0x fertility level. The other thing is, of course, you're letting real grazing animals going in there. We're clipping and basically emulating like a hay type.
We'd have liked to have done that, but the reviewers said we had to grade.
I was told. Yeah. So.
Sort of different question about the soil microbiome. Just wondering if you've thought much about, like, I mean, you talked about microbial growth, and that was important to soil carbon. Do you think that who are the soil microbes, like the fungi versus bacterial composition, would matter?
I suppose it would. I would have to go consult with my soil ecology friends and check in with, maybe that'd be a great question for Francesca, actually. What her current thoughts on that are. I don't know. Because I've heard both arguments. Sometimes you hear like, oh, fungi are where it's at. And you're like, no, it's the bacteria that have the most anabolic potential. So I don't know if that's rolled out.
Any other questions? Anything online? Okay.
Oh.